Let's talk now about the photoreceptor physiology. And what do we mean by photoreceptors physiology is to understand how those rods and cones respond to different amount of light and therefore making us able to see. And the first thing we need to establish in the rods and cones that their sensitivity to light, and this is something we already talked about, is different. Both of them will have the same type of pigment, and this pigment is responsible for or sensitive to light. This pigment is called rhodopsin. So in both in rods and cones, they have rhodopsin. But remember, we said rods are for dim light, and in cones are for bright light. What does this mean? It means that rods are very sensitive to light. Why? Because the minimum amount of light, when it hits the rods, it makes them to induce an action potential. Where our cones, they need a bright light. So they need much more amount of light to have an action potential. So those have less sensitivity or insensitive to light. But again, both of them would have rhodopsin inside them. Now the rhodopsin itself, the sensitivity to light is the same. So how do we understand this? I need you to think of the rods and cones as boxes. So we have rods and we have cones. I'll put it in different color. Not only in different color, but in a thicker line. So for the rods, when the light hits them, even if it's the tiniest amount of light, it will activate rhodopsin. So rhodopsin will get activated or broken down, and we will talk about it. So the minimum amount of light can pass through the rods. But the cones, since we have like thick wall, think of them like thick curtains, we need much stronger light to pass it through, right? Because if it was just tiny light or like not really strong light, very likely it will be blocked, right? So it is the same rhodopsin, it is the same pigment. What the difference is how thick this wall. This is not really accurate 100%, but for simplicity, we would just say it is about the walls of those in the cones or in the rods. This rhodopsin, when it gets activated by light, it turns between two states. Here, you can see it that, for example, when there is light, it will move from this position or a transretina to this position to the 11th cis retina. So this is in light. And when there is no light, it will go back to here. So it has two positions that it is switching between 1 and 2. We will call it like it will move from position A, let's say position A, or situation A or shape A, okay, into shape B during light. But then when there is no light or there is no light hitting this particular rhodopsin, it will convert back to A. In another word, to understand this, we would say again that for the rhodopsin, it has two shapes, A and B, and it switches between them. If there is light, okay, we will say that rhodopsin would get broken from shape A to shape B, broken. In case there is no light, it will regenerate. It will regenerate, or we'll call it accumulate. And this again happens in both, in rods and in cones. When light hits them, whether it's dim light or strong light, it doesn't matter. Rhodopsin will switch from A to B, or will get broken from A to B, and when there is no light, it will get regenerated and accumulated. And this means that it will go back from shape B to shape A. Now, what we need to understand is the light to dark accommodation, and also we need to understand the dark to light accommodation. What does this mean? It means that here we are in a bright light and suddenly we are moving to a dark light. Think, for example, that you are in the living room going to the bedroom without turning the lights on, right? And here, dark to light, it means that we are, for example, at night, we woke up at night and the bedroom, it's dark, but we are going to the bathroom and we turn on the light. So that is the dark to light accommodation and light to dark accommodation. And to understand them, we need to analyze the situation of both rods and cones and their rhodopsin in those two conditions. So let's start with the light to dark accommodation. So I will say light to dark accommodation. And again, this means that we are in a bright light moving to a dark light or dim light. So in bright light, what do we have? We have the cones are active because it's strong light. What does it mean for the cones? It's active. It means rhodopsin is constantly changing between A and B. And this means that rhodopsin is constantly sending, or because of this change of between A to B, we are constantly inducing action potentials in the cones, right? So this is in light. I'll put it here in green, in light. So in light, the cones are active, which means rhodopsin is moving between A to B, right? And that means that we have action potential. What about the rods in light? Still in light, okay? The rods are inactive. Why? Because it's too much light for them. What about the rhodopsin in the rods? In the rods, it is broken down. Why? Because there is always too much light that whenever it tries to build up, it's immediately broken. So it will remain always broken because it's too much light for the rods to accumulate or to build up any rhodopsin. And therefore the rods during the day or during the bright light, they are inactive. They don't send any action potential. Now let's move to in dark. What will happen to the cones? In dark, they will turn inactive immediately. Why? Not enough light to break down rhodopsin. Therefore, no action potential. No more action potentials. Right? But for rods, now it's perfect. Rods, they are fine in dim light. So they will start to get activated. So now they are active. However, to get them active, it means that their rhodopsin needs to build up or to regenerate it. To be built up or regenerated, or if you want to get accumulated, whatever you want to call it. The point is that the rhodopsin needs time. So in this case, until we get this time, until we get the rhodopsin accumulated, until that time, rods are not sending action potential. So this means that there is no signal coming from the rods or the cones yet when we immediately switch to dark light. So there is no action potential coming from either of them. So during light to dark accommodation, what we will see darkness, no vision. Why? Because cones suddenly turned off, and rods, yes, we turn them on, but they need time to restore their rhodopsin to be able to send the action potential. So this time sometimes will take like maybe 3 minutes or 2 minutes, or maybe sometimes 5 minutes, right? During that time, then there is no action potential coming from the eye or from the photoreceptors. That's why you see darkness. Now let's talk about the other way, dark to light. And again, let's talk about what happens in cones and in rods. In dark, let's start with the rods. Rods are active. It means they are inducing action potentials. Rhodopsin in rod, it means it's switching between A and B, no problem. So it gets broken and reaccumulate, and therefore we will get action potential. What about cones? This is in dark, okay? In dark. Cones in darks, we said those are inactive. Why? No enough light to induce action potential, or technically to break down rhodopsin. What does it mean there is not enough light to break rhodopsin? If you remember, rhodopsin, if there is no light hitting it, rhodopsin would accumulate. So now in the cones, we have plenty of rhodopsin. And since the cones are like thick walls, we said, then rhodopsin can easily accumulate. But yes, it's accumulating, but there is not enough light to break it down, therefore we don't get any action potential. In dark, that's in dark. In light, now we have bright light. What will happen to rods? Rods, it's too much light, so they will turn inactive, and their rhodopsin is broken without the ability to regenerate. But let's see what will happen to the cones, or in the cones. So now in light they are active. What does it mean active? It means that rhodopsin can be broken. And since we said like cones accumulated already plenty of rhodopsin, then all of this rhodopsin, almost in all cones will get broken at the same time. What does this mean? It means if we are talking about 100 million of cones in our eyes, roughly that is the number in each eye, then we are talking about 100 million of action potential sent at the same milliseconds. And that will cause the vision center just to see white light or a splash of white light. So that's what we see when we move from dark to light. White splash, right? White splash immediately, and sometimes it's going to be painful because there's so much action potential is going to the vision center in the brain. And this again, it will take a few minutes, maybe 2 minutes to 3 minutes, to get those cones, working at the regular speed. It means that breaking and regenerating rhodopsin, and therefore continue with the action potential. Okay, so that's what explains the accommodation from dark to light and light to dark, and explains what do we see each time we do that. Now, for the color blindness, we understand that the cones are the one responsible for the colors. Sometimes we are missing, or patients with color blindness missing one or more of those cone color pigments. So the most common type is the red or green. So either the red or green is absent, and therefore they will not see those colors. Here are different visions of different treatment. Here are different sites of different types of color blindness. Sometimes they are using, losing the red, for example, here, so they will not see really like red color. Here they are losing the green, for example, they will not see the yellow or the blue. And here they will use, or here they are losing the 3 different types of colors, so this will see just shades of black and white. You don't need to know the details of those conditions. Just understand that the color blindness is because absence of those cones. The last few things in this video are about the depth perception. This is how do we understand depth. This is an act of mainly the brain and the signals coming from each eye. So the brain will understand that if an object is large comparing to a known object that is also large, so our eye, if you put your finger in front of a tree and looking at your finger, you will understand that you will see that your finger is bigger than tree. But your brain understands that this is not correct, that your finger is not as big as a tree. So therefore, it will interpret that your finger is close to you versus the tree is far from you. So the size of this object that we are looking at will determine if this is close or far, and that's how we understand that there is a depth in the vision, right? Then this is one thing. Then the other clue that the brain would use is where is that object or the signal, where is it coming from? And as you can see, like there is a field of vision that both eyes will see, but also there is a field of vision that only one part of one eye will see it. So for example, this is the right side. The extreme right will fall on the will be seen by only the right side. And the same thing, the extreme left will be only seen by the left side. So if there is an object coming from here, like if there is a person standing here, and those cones or rods are sending that action potential, and this action potential reflecting this man or this object is coming only from this direction, this is how the brain will understand the position of the object. If both, like let's assume that there is a person here, and in this case, as you can see, this is both eyes will send those action potentials. So the person, or the brain will understand that this person is in the center. So this is like the signal, how the signal is coming from both eyes. However, although this is kind of a side information, the brain is handling the information coming from the left side of the body. They will be all handled on the right side of the brain, and the information coming from the right side of the body will be handled by the left side of the brain. Why is that? Because we have the optic chiasm, and this optic chiasm is the area where the optic fibers from the optic nerves would cross over to each side. So that is like reorient or redirect the signal coming from each area within our eyes. So that ends the eye. In the following video, we'll start talking about the ear.